All right, so very briefly, this is where we stopped last time. Okay, we're discussing about the general idea in chemistry of what is stable and what is not stable. Okay, so again, you should always remember, if a chemical is reactive, that means that it's less stable than its product. All chemicals in nature, essentially, you can view it as a relative thing in terms of their stability. Okay, any chemical reaction, you always have reactant and product. Okay, so if the product is more stable, okay, that means that the reactant is, relatively speaking, less stable. Okay, so it's more reactive. Right, so same logic applies to acids. Okay, so if you have any given acids, whether organic acids, inorganic acids, we just say HA. Where A is the acid chemical, and then H essentially represents a proton. Okay, and any acids can dissociate. Okay, you pass the proton to water. Okay, so that defines in our case this thing's an acid. All right, now for any acid, or actually for any proton, okay, any proton, there is a pKa for each individual proton in a chemical structure. Okay, and the pKa is defined as the negative log of Ka. Okay, and Ka is just this expression right here. Right, so it tells you how likely it is for your proton to move away from your molecule and end up on water. Okay, so that's what pKa means. Lower the pKa means that the more likely, okay, that this proton will end up on water, will end up elsewhere. Okay, it's easier to leave. Now, so the question is then, how easy is the proton removed from a compound HA? Then the answer is, it really depends on how stable the conjugate base A- is. Okay, so for example, phosphoric acid, you have this in Coca-Cola, like the Coke that you drink. You got some little bit of phosphoric acid to it. That's why people say, stop drinking Coke. I'm just kidding, right? Phosphoric acid, it has actually 3 pKa's because it has 3 protons that we can pull off. Okay? Of course, every proton that you want to pull off actually make it less stable, okay, because you have excessive charge that end up on your conjugate base. Okay? So the first proton you pull it off, you get H2PO4 minus, okay, 1 minus charge. That's pretty okay. So the pKa, it's pretty low. And then you get to the H2PO4, 1 minus, becoming 2 minus. Obviously the pKa increases because it becomes harder and harder for water to take off that proton on the phosphate. Make sense? Okay? So this is a very key principle, okay, that we really need to remember as we later on talk about proteins, protein structures, amino acids, and that kind of stuff. All right? So pKa, again, does not only apply to acids, okay? It applies to any hydrogen that you can think of. For example, you can take an alkane. Alkane, it has a CH3 at the very end. That proton has a pKa, okay? It's pretty meaningless because the pKa is super high, okay? The pKa is about 50, right? So then the question you should have is, wait a minute, so is 50 a large number or a small number, okay? The way we judge whether or not it's large or small is you just compare it to water, okay? So here's water. Water has a pKa of 16. That means that in order to have your alkane hydrogen to be lost, okay, it's actually very difficult using water to take that proton. All right, so essentially that's not going to happen. Okay, in biochemistry, you do not really, at least in open aqueous conditions, readily take away protons from alkanes. You don't do that. Okay. So basically, these guys you don't really see. that much. Okay, they will not lose their protons readily. Okay, because they're very stable. So for our purpose, we are interested in these. Okay, these are the ones that are relevant to biochem. Okay, minus the sulfuric acid and those guys. Okay, minus sulfonic acid. So we're going to primarily be focusing on carboxylic acid, protonated amines, those two. Okay, so those two we will really always see because they're found on amino acids. Okay, they're found in various organic acids. Okay, so these things are, so this guy right here is an organic acid. Okay, carboxylic acids are, generally speaking, organic acids. All right, so those guys are the ones that we're really going to look at. Okay, and turns out pKa for, let's say, carboxylic acid. Okay, on the chart, the pKa says it's about 4. Okay, however, that changes. Why does it change? Right, it's a pKa. pKa should be a set characteristic of the molecule or of the proton. Why does it change? Okay, pKa can actually change depending on the environment. Okay, for example, if you have, later on when we talk about proteins, let's say that you have a organic acid right here. Okay, and next to it you have other charged molecules. Okay, those charged molecules will affect the pKa of this proton. Okay, that's how it is. Okay, because it will make it stable, more stable, or less stable, depending on the charge surrounding that particular functional group. 反正这边主要跟你讲说，carboxylic acid 很重要的原因是因为它的 pKa 其实会不太一样 ，depending on 你在什么样子的 environment。因为如果你今天这 carboxylic acid 就只有它在水里，它可能 pKa 是一个 value。但是如果这个 carboxylic acid 呢出现在一个蛋白里面，比如说蛋白里面的一个 R group 身上有一个 carboxylic acid， 那蛋白很大嘛，那如果那个 carboxylic acid 附近有其他的 R group 也有 charge， 或者是有一些 polarity 的效果的话，它会让那个 pKa 会变。OK， 但是不可能从4然后变到8。但是可能会是4到2，然后或者是在中间跑。OK， 那个对我们来说就很重要，因为我们在 study enzyme mechanism 的时候，我们就会看到。OK。Alright, so that's all I have on pKa's and the general review for chemistry that you guys should know. Okay, so that's the level of chemistry that you definitely should know. Okay, so anyone not comfortable with Ka's and pKa's? Okay, so for our purpose, just please remember the lower the pKa, the easier it is to remove that proton. Okay, so that's really all you have to remember. All right. All right, so let's talk about evolution a little bit. As scientists, maybe less engineers, but scientists really always have one central question that they want to answer. Okay, and essentially that is the origin of life. All right, so scientists across the world always try to get the answer to origin of life. So a while ago, in 1953, there was this experiment done by Miller and Urey, and essentially what they did was they tried to simulate prebiotic world. Okay, so this is their apparatus setup shown on the left right here, and essentially they have a little reservoir of water. Okay, just water. And the water is trying to simulate ocean. So as you heat up the water, the steam, okay, is going to travel through this tube and end up in this vessel right here. And inside of this vessel, they also put some other inorganic gases such as ammonia, methane, hydrogen, that kind of stuff. Okay, and then they put electrical charges, okay, electrical spark, to simulate lightning. Okay, sure. And if anything that reacted, okay, if anything that reacts and you produce any product that is no longer a gas, okay, and essentially will sort of come down here, okay, and you got a condenser that traps it and make it into the liquid phase, and essentially that kind of stuff is going to end up here in this trap. right? So then every now and then they would open this sampling probe to take a look at what's inside, right? And what they can find is, surprisingly, you get some amino acids, hydroxy acids. So hydroxy acids are acids with OH, and the OH can be anywhere, hydroxy acids, etc. Right? So they can show that maybe this is how the world was able to generate some organics, okay, in the form of amino acids and hydroxy acids and that kind of stuff. And of course, in order to have life, okay, you kind of need these small molecules. Okay? So this is sort of a very simple experiment, but very interesting experiment that showed potentially going from these very small gases using high energy charge right here, and then you can get some complex, well, not complex, but more complex molecule. This is something that you will always see, you know, you saw this back in middle school, no, it's probably high school, okay? Yeah, you saw this in high school, you saw it in general bio, yeah, you're going to see it in biochem, okay? So the idea is very simple. This is what we call central dogma. That is the understanding of how life sort of moves on, okay? So you get DNA to RNA, RNA to protein, okay? So you got transcription, translation, right? That's great. However, okay, this is the current understanding of central dogma, right? So central dogma would say DNA makes RNA, RNA makes proteins, okay? However, it turns out, okay, really RNA is sort of the universal thing, okay? It's like the universal thing. Why? Because RNA makes protein, we know that, okay? And turns out RNA can also make DNA, okay? Through this enzyme called the reverse transcriptase, right? And what that will do is RNA, it's also a template, okay? And you got this enzyme that's going to help you, based on this RNA template, match with it and make DNA, okay? So RNA can go back to DNA. So this is the current understanding of central dogma, right? And RNA can also make RNA as well because it's a template, right? Now then the question is really, okay, that's interesting, but then which came first, DNA, RNA? Did RNA evolve the function to go back to DNA, or did DNA evolve from RNA? So that's sort of a question that people like to ask, right? Now the answer to that is really the current understanding is that most likely RNA came first, okay? And one of the reasons people believe that is actually because RNA is capable of maintaining information, so that's the function that you guys know, and catalysis, okay? So there are these things called the ribozymes, okay? Ribozymes? Sounds kind of funny. Yeah, so those are RNA catalyzing things, okay? So they are RNA molecules that are capable of catalyzing reactions, okay? That's cool. So even capable of forming peptide bonds, okay? So that tells you something that potentially through evolution, okay, this is where the evolution showed that catalysis was initially found on RNA, and having RNA capable of catalyzing reactions, then gradually they can generate proteins. Make sense? Right? So then through evolution, eventually the catalysis function went to proteins, okay? And the information conservation function went to DNA. Why? Because shows here protein, you got 20 amino acids and X in terms of length. Okay, you can make it as long as you want. So basically protein gives you diversity in structure, okay, and function. Because when we get to amino acids, we'll talk about all the 20 different amino acids, okay, and how they have different properties like hydrophobic, hydrophilic, aromatics, charge, that kind of stuff. Okay, so all kinds of properties happen on different amino acids. Okay, and you have 20 different kinds of amino acids. So let's say if you have a protein that is 300 amino acids long, then you have 20 to the 300 power in terms of possible structures. Okay, so that's huge. And then the information conservation went to DNA. Okay, that is because DNA is more stable. Okay, and one of the reasons why it's more stable, okay, by looking at the structure, this guy is RNA, ribonucleotide. This guy is DNA, which stands for deoxyribonucleotides. Okay, is the loss of a hydroxyl group on the 2' carbon. Okay, we'll get to the naming when we talk about, not naming, the numbering when we talk about sugar chemistry towards the end of the class. But essentially, on the 2' carbon, this is the second carbon, you lost the OH. Okay, and because you lost the hydroxyl group, it can form less hydrogen bonding, right? Make sense? You form less hydrogen bonding, you have less structures, okay? Less possible structures, all right? So basically then RNA, okay? So DNA is just my ugly drawing of double helix, okay? RNA, when single-stranded, okay, you can make all sorts of funny-looking structures. Okay, for example, they can look like that. Okay, and structures are essentially the most important feature in biochemistry. Okay, 这个很重要，所以用国语讲一次 structure 3D 结构。Okay， 对于 biochemistry 来说，大概是最重要的一个 characteristic. Okay, why? Okay, we'll get to catalysis. We'll get to the Y part towards the catalysis, but essentially you can imagine this, okay? Let's say you have two chemicals that want to react. Okay, let me draw it. You have two chemicals, okay? Chemical 1, that looks like that, okay? Chemical 2, that looks like that, okay? If they want to react and become this, okay? Let's say they have to react and become that by forming a bond, okay? What has to happen, right? They have to, first thing is get close together, right? They have to, in order to form a bond or in order to react, they have to physically be next to each other, right? Physically next to each other, okay? So they have to get to each other, and guess what? The orientation also matters, okay? So if they're going to react like this, you cannot have it like that. This will not react, okay? So orientation and direction also matters, okay? So we agree. Now, then the question is really, okay, if you don't have something that helps it orientation to be like this, okay, your molecule is randomly just rotating around in solution or in whichever medium they're in, okay, they're just rotating, okay, and the chance of them hitting each other in the correct direction is probably going to be low, okay. So what enzymes, okay, essentially what enzymes does, okay, what enzymes do is it helps you to force these two, okay, to be in this direction next to each other. That's what enzymes do, okay? So enzymes, essentially you got one big ass enzyme and it comes in and it grabs your reactant molecules and physically put them together, okay? That's what enzymes do, right? Now, once they're physically close to each other, then your molecules can react easily because orientation and proximity is already there. 这很重要，这边先跟你讲一次，然后那个 towards 大概第2或第3个 part 会再跟各位再仔细的讲一次哦。 这边要跟各位讲，为什么3D 结构很重要 ？OK， 这个很重要。OK， 原因是因为你今天有2个 molecule， 它们要 react。OK， 那它们要 react 的时候，方向跟距离一定要对。OK， 那不对的时候，它们就只会不断的撞，就一直撞，直到撞到那个正确的 orientation，and then 它们会 react。OK，enzyme 呢，今天是一个3D 结构，它会把你的2个化合物接在一起，就是吃进来。然后把它们 literally 放在 each other 旁边 ，OK， 然后让它们去反应。So enzymes can do that because enzymes have a 3D structure. Okay, now this also shows you why RNA can do the same thing because RNA also can form complicated 3D structures. Okay, so that's why RNA can also help to catalyze some simple reactions. Okay, but of course enzymes do much more than that. It can also change some stuff about the molecules, but we'll get to enzymes towards the end. Okay, so this is important, right? Structures of your big molecule can help your small molecules to react. Okay, so that's the one of the fundamental features of biological catalysis. If you print out my PowerPoint, you can take a look at this. This is what people drafted as a hypothesis for a possible RNA world scenario. Okay, so essentially it shows what kind of steps are necessary to then form the life that we have right now, starting from very basic stuff such as nucleotides. Right? So it says creation of probiotic soup, including nucleotides from components of Earth's primitive atmosphere, like what we saw in the Miller-Urey experiment. Okay, and then towards that, you got some small chances of production of short RNA molecules with random sequences. Now, the key is, of course, random. Okay, and having random RNAs, and of course, RNAs, some sequences are good in a way that they can self-replicate. Okay, they can make more of the same RNA. And then through random chance again, you start to be able to synthesize, okay, RNAs that can catalyze peptide bond formation. They can make proteins now. Okay, and then as you make proteins, gradually you have an increasing role of peptides in RNA replication. So the proteins that you made actually helps, okay, RNA to evolve, sort of. Okay, so you got a co-evolution of RNA and protein. All right, and then you got primitive translation system that develops, okay, with RNA genome and RNA to protein catalysts. Okay, so as you can see, gradually you're making more complicated systems. Okay, and then you got genomic RNA. Okay, and somehow you evolve the function of genomic RNA to DNA. Okay, and then gradually, because DNA is more stable, DNA continues to be the information storage. Protein becomes the catalyst. Okay, so this is sort of a possible RNA world scenario that people thought. Okay, all right. So how big are these things? Okay, so a lot of times in biochemistry, what you notice is that sometimes we don't have the same numbering system or the same nomenclature as organic chemists. Okay, but in general, this is what we're going to talk about in terms of biochemistry. Right? DNA from one chromosome has a mass of about 20 billion daltons. Okay, daltons. So here is a very important unit definition called dalton. You probably learned this back in organic or general chemistry. Your professor probably just said, know this. This is a unit. Okay, and dalton is really nothing more than just 1 gram per mole. Okay, it's 1 AMU. It's just a different name. Okay, now why is this important? Okay, because in biochemistry, enzyme size. Okay, size. We usually say, oh, your enzyme has a size of 30 kilodalton. Okay, or your enzyme has a size of 90 kilodalton. Okay, so when we talk to fellow biochemists, Daltons are the units that we use. Okay, so you have to get used to that. Right? And this is important also for a variety of things. For example, for your undergrads, okay, in our department, you guys are probably thinking about going to grad school later on, okay? And graduate school in Taiwan, you got two choices, right? You can take an entrance exam, okay? But of course, you guys don't want to take an exam, so you can go through the application route. Okay? You can apply, okay? Apply, and for that, you have to take a oral examination, sort of, okay? You have an oral exam. And for that oral exam, you're going to talk about what you did as an undergraduate researcher, okay? And you're going to say, blah, 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 I did this, I did that, okay? And I bet half of you will be working with proteins, okay? Proteins. And then you say, oh, I studied this really cool protein because I can do this, I can do that, all right? And then your professors or the examiners, okay, I am one of them, and we will ask you a question that, okay, how big is your protein? Okay, and you're going to tell me, my protein is 35 kilodaltons. Okay, that's good. So far, so good. And then another professor is going to ask you this question. What unit is Dalton? Okay, and this question actually killed about half the students. Why? Because they don't remember Dalton is a unit for mass. Okay, it's actually a unit for molecular mass. Okay, and professors will always ask and say, okay, so Dalton, is it a unit for length, size, weight, what? Okay, they can't answer that. Okay, so then of course they don't get into grad school. Okay, so for you guys, definitely you need to know what Daltons are. Okay, so this is very important. Right? So proteins. Protein molecules typically have a mass of 10 kDalton, okay, to about 1,000 kDalton. Okay, we don't usually say just Daltons because usually we always put a little kilo in front of it. That's make the number smaller to deal with. Right? So proteins usually is around this range. Okay. Anything larger than a couple hundred Dalton or kDaltons is considered very large. Okay, but we'll get to proteins in a couple weeks. Okay, so that's that. This is sort of a random definition that I want you guys to know. All right, so we're going to now talk about Hochland's 7 pillars of life. So back in, I think it was the 1980s or 90s or early 2000s, something like that. So this professor from UC Berkeley, sort of he's, you know, as you study more about biochemistry, you notice that a lot of these scientists, they eventually develop into philosophers. Okay, they really think about like origin of life and what constitutes life, that kind of stuff. Right? So then this guy, essentially, he came up with 7 features, sort of, or 7 characteristics that you need to have in order to form life. Okay. So the first thing is program, okay? And what he considered program is really an organized plan for the constitution and regeneration of organism, okay? Essentially a blueprint. So for our purpose, it's DNA, okay? So it's like a genetic blueprint to store such a complex information, okay? So that's the first thing you have to have, program. Second, improvisation. So this is the ability of living matter to change the program to assure survival as the surrounding change. Okay. Now you got different bacteria that grow on your feet, that grow in your stomach, that can grow in hot springs. Okay. What do they have in common? Not much. Okay. So what they can do is they can adapt to specific environments. Now this is a permanent adaptation, not a transient adaptation, okay? So they are capable of evolving, evolution, okay? Evolving to survive under those different conditions, okay? So that's important. You need to be able to change your program to adapt to your environment. And then 3, compartmentalization. This is the ability of an organism to separate itself from the environment. okay, such as with membranes. So obviously this is important because you need to be able to tell what's you and what's not you. Okay, you as an organism, you say, okay, so everything in me is me, okay, and everything outside is the environment. Okay, so if you don't have a cell membrane, your proteins, your DNA will leak. Okay, so obviously you don't have life. You need to be able to draw a little circle and say, this is my personal bubble. Okay, you don't touch it. Energy, I don't know why I show you this guy. I think it meant to show that the living matter must create levels of complexity. Okay, so you need to be able to sustain a complex system such that you generate your program. Okay, you have to have the ability to get energy from something to maintain your program. And then you got regeneration. This is the ability to compensate for the inevitable wear involved in maintaining a physical state far from equilibrium. Okay, so again, in biology, we don't deal with equilibrium. Okay, we actually deal with homeostasis. We'll talk about that in a little bit. Number 6, adaptability. This is the capacity of an organism to respond to environmental change. This is kind of different to number 2. This is sort of a transient adaptation. Okay, what does that mean? For example, in Taiwan, we get in the winter temperatures about like 15 degrees Celsius, but then in the summer you can go up to 35 degrees Celsius. That's a change in environment. Yeah. Okay. You have to be able to adapt. Okay, adapt to that kind of environmental change, temperature change. So let's say there's an organism that can only grow at 37 degrees. Yeah, that organism cannot possibly move on. Okay, because if the temperature goes above 37 to like 38 and it dies, to 36 it dies, then it's too easy to die, such that that organism will not pass the DNA. Okay. Number 7, seclusion. Okay, seclusion. This is the metabolic processes and pathways must operate in isolation from one another, even though they may take place within the same compartment of the cell. Okay, so this is sort of like it's like you got all these weird chemical reactions happening at the same time. They don't really interfere with each other. Okay, that's important because in order to have such a complex organism, you have to have complex chemistries, okay? But you don't have individual compartments for every chemistry, so you must be able to tolerate all that chemistry happening at the same time. Okay? Make sense? All right? So that's that. And then interconnected with the 7 pillars of life, okay? It's the function of semi-permeable membranes. So that's important because semi-permeable membranes allow things going in and out. Okay? If you only have membranes, okay, you have a permanent separation between the cell and outside. Okay? That would be bad. Why? Any thoughts on why that would be bad? A permanent separation between the cells inside and the cells outside. Why is that a bad idea? What would happen if you don't have transportation between outside and inside? So this is actually very simple. Right? Let's say you as a person, okay, you have to eat, right? You don't eat, you don't get energy, you die. Okay, same idea. You have to sort of pass things between your surrounding and inside the cell. Okay, that's just how it is. Because otherwise you have no energy. Okay, you have to be able to exchange material between intracellular and extracellular. Okay. Lastly, this is very important. Right? This is very important. Homeostasis. It's a condition in which the chemical composition of a biological system is held constant. This sounds awfully like equilibrium, but it's not. Let me show you what equilibrium looks like. Equilibrium means you go A to B, and then B, back to A. Okay? So this guy is an equilibrium. So the concentration of A and B are constant. So concentration of A and B is constant. Okay? However, this cannot happen in life. Okay? In biology, this do not happen. If you have equilibrium in you right now, you're dead. Okay? Why? Because you are actively generating heat. You're actively generating energy. Okay? Now, if that's the case, okay, how is it that your composition or the chemical composition of biological system is held constant? Okay? So homeostasis essentially works like this. You still have A to B. This is something that has to happen. Okay, let's say you drink alcohol and you need to metabolize it. The metabolism, or at least the metabolic pathway, go from ethanol to CO2 is always the same. Okay, so A has to go to B. Okay, now concentration of A and B still constant. How? Okay, so turns out you have a net input and a net output. Okay, so this is sort of what homeostasis means. Okay, A and B stay roughly constant. Okay, concentration don't really change that much. Okay, it can change, but it doesn't change very much. Okay, it remains relatively the same. However, you have a continuous conversion of A to B. Okay, B doesn't really go back to A. Okay? However, if you have a continuous conversion of A to B, how do you have more A? You have A from somewhere else. Okay, so life is always a continuous process. Okay, life cannot be equilibrium, because if life is at equilibrium, you don't net generate anything. You don't generate any net energy. If you don't have net energy, then, well, guess what? You're not alive. Okay? So that's important. All right? Equilibrium versus homeostasis. All right? So evolution goes, billions of years ago. Okay? This is sort of a rough time scale of what people think about when certain things happen. For example, initially you got some formation of Earth, okay, formation of oceans and continents, okay, and then you start to generate photosynthetic bacteria, not the oxygen generating ones yet, okay. Initially, it's just photosynthetic bacteria. They don't generate oxygen. We can talk about that later. And then you generate O2 producing cyanobacteria. Okay, cyanobacteria are the ones that do photosynthesis, produce oxygen. Okay, once they produce a lot of oxygen, you start to have aerobic bacteria, such as E. coli, that kind of stuff, things that can use oxygen to grow faster. Okay, so bacteria. And then you start to have eukaryotes. Okay, once you have eukaryotes are moving around, bacteria is moving around. Somehow they decided that they're going to be best friends, and then they're going to live together. And you got mitochondria, plants, that kind of stuff. So you start having algae. Okay, after you got algae, okay, you evolve further to form other eukaryotes like plants, fungi, animal. Okay, so that's sort of like a little timeline. It goes like that. Okay. Now, I always like a pictorial view. Okay, like pictures is always good. So initially, you start with some kind of anaerobic eukaryotes. That was like the first eukaryote, whatever. Okay, and what separates eukaryotes from prokaryotes is that your nucleus has DNA. Okay, so you got two membranes, kind of. Right, and then what happens is you decided that, hey, I too lonely, this guy is lonely, okay? And then it wants a friend, aerobic bacteria, okay? So eukaryotes decided that it's going to eat the bacteria, and then so bacteria comes in, okay? And they sort of just live together, okay? And you get your aerobic eukaryote. That's fine, okay? Eukaryotes, now it's aerobic because it has mitochondria. This is why, okay, we think, okay, of course nobody can know this for sure, right? Because this was like billions of years ago, so nobody knows. We think that's why, okay, mitochondria has its own DNA. 知道吗? Right? Mitochondria has its own DNA. And that DNA looks a lot like bacteria. Okay, so people think that's how it happened. And then, out of nowhere, this happy eukaryote decided that it's going to eat another thing called cyanobacteria. This guy can do photosynthesis. Okay? And it grabs the cyanobacteria. Now you got chloroplast. Okay? So it turns out photosynthetic eukaryotes like algae and plants, they have both mitochondria and chloroplast. Okay? Even though people was like, well, why? But they do. Okay?